Well, thank you to the organizers for having me. It's really a pleasure to be part of this event. So hello to everyone on the web and to all you guys in the room. My name is Kevin Olival. I'm from EcoHealth Alliance. For those of you that aren't familiar with our organization, we're a nonprofit research organization based in New York City, but we do international work, a lot of work in Asia, but kind of all around the world, looking at the connection between ecology, human health, and wildlife health. And so actually our roots began as a wildlife conservation organization, but we've been moving into the health space over the last decade or so. So a lot of what we do at EcoHealth is sort of think about the big picture, the trends, the patterns, why are diseases emerging, where are they emerging. So I wanted to start, I know the focus today is on Ebola, but go sort of a step back and talk about disease emergence in general, which I will be sort of interspersed throughout the talk. But this is a paper that we published in 2008 in Nature, Kate Jones and others, and it shows sort of very convincingly that emerging infectious diseases have increased over time. So this is sort of data on a per decade basis from 1940 to 2000. And this is basically by trolling through the literature and compiling a data set of 330 or so emerging infectious diseases. So these are the events where that disease first emerged, or we first discovered it, right, emerging into the human population. And so if you look at this bar chart, you'll see that most of these, sort of everything above the black bar, come from animals. They're zoonotic diseases, right? It just means a disease that comes from animals. And of those zoonotic emerging diseases, 72, about 3/4 of them come from wildlife. So there's a very important role for wildlife in understanding disease emergence and sort of predicting what the next pandemic will be if we're able to get there. Process, but we can actually break it down into some pretty simple steps. And so this is a paper that was published in Lancet last year. And if you look at, there's basically 3 levels of emergence. And the first one is really this pre-emergence stage, which is when these pathogens, as I said, 3/4 of them come from wildlife. There are viruses and bacteria and diseases out there in the animal population. Well, sometimes those are shared amongst the animals. Sometimes they jump over into a new species, right? We might call that cross-species transmission or spillover from one species to another. And sometimes that other species is humans, right? And so at this sort of initial stage are these drivers, we call drivers of emerging diseases, which are things that humans are doing to the environment. So we are cutting down the forest. We are hunting animals from the forest. We are intensifying our livestock production in a massive way, right? All these things are drivers for emerging disease. Well, then you get these sort of periodic little bumps and spillover where a disease may jump into the human population. Sometimes it will just burn itself out, and sometimes it will take off and become a large epidemic, right? So you kind of see these green things where they're just like maybe a few cases jumping over and spilling over into humans, and then sometimes you see like we're Ebola in West Africa right now, you see a large epidemic happen. And then how does that go pandemic? Well, that's really an issue of global travel and trade, right? And we can model this, we can predict the spread, and there's been some nice papers that have come out for Ebola sort of predicting the spread of where it's gonna go. Well, you can take airline data from around the world and it's a numbers game, right? So how many travelers are going from point A to point B? And then you can look at sort of predicting pandemic spread based on those data sets. And we're definitely living in a much more connected, globalized world today, so it's no surprise that it's easier for pandemic emergence to happen when these things were previously sort of burning themselves out in maybe more remote human populations. So the pathogen needs no introduction. We're here to talk about Ebola virus today. But Ebola virus is a filovirus, the family Filoviridae, which is the Latin word for phylum or thread. So when you look at it at 150,000 times magnification, if you happen to have a transmission electron microscope in your room or your house or your dorm or your lab, then you could look at things like this, and that's what it looks like. That's the creature we're dealing with. But Ebola virus is not just one virus. There's actually several Ebola viruses. So there are actually 5 Ebola virus species, and I'll talk about that in a minute. just give you a sense of how they're related to each other. And this current outbreak, and this figure is from the New York Times, and this is now a couple of months old, so it's that bubble's a lot bigger now in West Africa. But it shows you the number of human cases, and this is sort of through time, from the 1970s to present, right? So what you see are those little red spots, mostly in Central Africa, are other spillover events, essentially, other outbreaks that have happened of Ebola virus and Marburg virus, other filoviruses. So we're certainly dealing with the most recent and the largest and the scariest of these outbreaks, but it is a disease that has jumped over into the human population a handful of times. And there's a lot of interesting things going on there, but I think I just want to make the point that, this is not, as I think you all know, the first time it's happened. And so what are the factors that cause these diseases to jump in the first place, right, is one of the big questions, and that I'll be addressing today. And not everyone, this hasn't been that widely publicized, but there was a separate spillover event of Ebola this year in DRC. And it affected about 80 people, but it's been very successfully contained, right? So separate event, not because of someone traveled from West Africa to DRC, it was actually a separate spillover event that the government in Democratic Republic of Congo dealt with very rapidly and effectively and have contained. So this is the phylogeny of the filoviruses, and I'm not going to go into too much detail on it, but I want to just kind of point out some geographic aspects. So Zaire Ebola virus is what we're dealing with now. That's the strain that is causing the current outbreak in West Africa, right? And you see right here, there's a little star, but a lot of the cases have previously come from Central Africa. And these are kind of just rough localities. There have been multiple outbreaks of some of these. And then there are two other Ebola virus species, if you want to call them that, that are sort of highlighted in purple there, that also jumped into the human population in Central Africa. And Marburg virus, which is Ebola's ugly cousin, you probably have heard of. Another very deadly virus, has had several outbreaks, including one in South Africa. And there's this strain that actually only caused one human case called Thai forest Ebola virus, and it did not kill that person. It was actually a veterinarian doing a necropsy on a chimpanzee, was infected, but did not die. So then we found a new strain from West Africa, and this is several years ago. But interestingly, and this is where I think it gets really interesting, is there's now these new filoviruses that we've been finding in other places, right? So there was a big die-off of bats, Miniopterus sherbersii bats, that's the species, in Spain. And they did an investigation of this bat die-off in Spain, and lo and behold, they found and they sequenced the genome for a new filovirus called, I never know how to say this, uveo virus. It's a quavivirus, so it has a new genus, so it's actually this whole new genus in the family. So, and then we have this other one, Reston Ebola virus, that we didn't find out about until it came to the US, to Reston, Virginia, because we were importing primates in for laboratory studies. And lo and behold, they had another Ebola virus called Reston Ebola virus. And we figured that out in the lab in the US, right? But then you go back to the Philippines and there's evidence that Reston is not only found in macaques, in primates, but also it got into the pig population. So it got into the agricultural system in the Philippines as well. But it's not very lethal, so we're lucky there. Maybe we dodged a bullet. Maybe it has the potential to evolve and become lethal in the future, but it seems like it's not a very lethal human pathogen, but it is another of these strains of Ebola, right? And it's way out there in the Philippines. And there's some evidence from pigs in China as well. So recently I published a paper with my colleague David Heyman, and this is open access, so if you guys want to download it, I like to think it's a nice summary of what we know about the ecology of Ebola so far. And especially focused on bats. What do we know about these viruses in bats? So spoiler alert, bats have probably a key role here. And I've done a lot of my research on bats, so I will talk to you a little bit about them. But the figure, and this is a figure from our paper, I think really just highlights how much we don't know about Ebola virus ecology. So we know that some species carry Ebola, but we don't really know how they're spreading it amongst different species in the wild, and even how many people get infected. So many of the outbreaks, we do not have we can't pinpoint the animal source specifically. A lot of what we know has come from later investigations where we've gone out in the field, the sort of scientific community, and have looked for these viruses in the natural system. But there's a lot of work that we can do, and we highlight some of the ideas in this paper about future studies. But one is certainly just understanding what are the species that are the most important in the cycle. We have some clues, but it's definitely not definitive. How are people getting those viruses from these animals? Again, we have clues, and I'll talk about those. Are bats and primates sharing the viruses in the wild? Probably, but how, right, is another question. And hopefully we'll hear some of this later when you hear more about primates later in the session too. But from a bat's perspective, I think there's definitely a lot of interesting things going on with the ecology of Ebola virus. So these are 3 fruit bat species that we have pretty solid evidence for that they play some role as reservoirs for Ebola virus. I'm being careful with my words because there are likely other species involved, right? But these are 3 that we've gotten nucleic acid from the animals. So we've gotten PCR, and that PCR is Ebola Zaire positive. And we also have serology, right? So you can also do a serosurvey in the wild animals, and you can find out if they have antibody evidence for past infection. So serology is good for sort of identifying at a population level, have these animals had exposure to these viruses before, but there are issues with cross-reactivity, where you can't really pinpoint exactly what virus it is, but you know that the immune system is reacting to something Ebola-like, at least, right? So these guys from work that Eric Leroy had done in 2005 we know carry Ebola in the wild, right? These are wild-caught animals where they did these surveys. And these are all frugivorous bats, so they feed on fruit, and that's all they eat is fruit. Fruit and nectar. Many bats in the world, about 70% of them are insect-eating bats, right? But there's sort of 2 big groups, and these are fruit-eating bats. Pretty cool animals, actually. Pretty impressive. And some of them have a capacity to fly great distances, right? Some of them are migratory species. So this is a really nice paper that came out earlier this year, not by our group, but by Pigot and others. Sorry if I butchered his name. But what it shows, and I think this is a really cool study, it's ecological niche modeling is the tool used here. So they basically looked at, first of all, where past human outbreaks have occurred from Ebola virus. Then they looked at the distribution of these bat species that we know are positive for Ebola. And they basically overlay that with climate variables and habitat variables, and you create what is a predicted niche or a predicted distribution for Ebola. So this is sort of the predicted distribution, right? Where it's green, it's more likely you have the potential for Ebola virus to occur in that area. And where it's sort of yellowish and lighter colors, it's less likely risk, right? So you see this risk includes a band through West Africa, but also this big area in Central Africa. And that is really an ecological tool, right, that's now used as sort of a disease prediction tool, approximation. So at sort of a crude scale, at a 10 square kilometer scale, we can make these predictions. What I think is interesting is then overlaying what are people doing, right? And this is another study not from our group, in PLOS Currents, which is really cool. They basically took human migration data and they mapped that out. And so the darker oranger areas are areas that are highly connected, where human population is highly connected. There's a lot of movement, there's a lot of people are connected, and they actually use cell phone data, so they actually use like people's movement records based on cell phone data. And then the green areas are areas with really low connectivity. So if you look at parts of Central Africa, there's actually quite low connectivity. So why is the current West African outbreak so bad? And I think we'll hear more about the epi later on, but just a suggestion, right? Well, it emerged in a place that's highly connected, so that helped it to get to cities more easily. Whereas the black dots are past Ebola outbreaks, were in areas generally of lower connectivity with the human population, right? So if you think back of the the niche model of where the hosts occur, where the wildlife reservoirs occur. They occur in this area, but there's a very different human landscape across that area too, right? And it's not just where people occur, but what are people doing? And we can get more into I think the role for anthropology and sociologists is really trying to understand what are people doing on the ground as well. That's putting them in contact with animals and the behaviors that they're specifically doing when they come into contact with animals. So we often get out there and actually do on-the-ground investigations of the origins for different zoonotic diseases. And I've been fortunate enough to be part of some of these studies with Nipah virus and with MERS coronavirus. I have not worked on the ground I worked on the ground with Ebola resistant virus in the Philippines, but not in Africa. But these pictures, so these are from different projects in different areas, but they show you some of the work we've done with bats, where we actually go out there in the field, we catch animals, we take specimens from those animals. We do not kill animals. We do this in sort of an eco-friendly way. So we basically collect a throat swab, a rectal swab. It's not very pleasant for the bat. We take some blood. So this is us pipetting just a tiny amount of blood from the brachial artery from the bat, and then we let the bat recover and we let them go. So what we're trying to understand is, how are these viruses being shed in animals, right? How are they being excreted and shed and potentially passed on to humans? We're not just grabbing animals and grinding them up to find viruses. We want to know what is the interface that they're having with people. So if we find the virus in saliva, well, you might want to avoid eating the same thing that those bats are eating, which is the case with some viruses like Nipah emergence in Bangladesh. So, and if you find it in the feces, well, you don't want to be hanging out where bats are pooping on you, right? So the sort of the route of transmission, what you can determine by this non-invasive sampling or minimally invasive sampling, is the route of transmission as well. So that's an important tool. So when we go out in the field, we also sort of set up this mobile laboratory to do the specimen collection, and we take biosecurity very seriously because we don't know what these animals carry. So, you know, even though there's a lot of wildlife biologists out there catching animals all the time with bare hands, we're out there trying to find viruses and study viruses. So we wear, you know, full protection depending on the circumstance. Just as you would if you were handling a human case, right? Because you don't know what the risk is. So we take the precautionary approach, really. So several years back, in this study that I was involved with, we published an EID, antibodies for Ebola virus in Bangladesh. And so this kind of got a little bit of a big splash just because it was the first time outside of Africa that we found evidence for Ebola in bats in a natural host. And now we didn't actually find the virus, we just found the serology. So we found antibodies to the virus in this population at a pretty low level. About 4% of the animals were positive, seropositive. But I think the importance of the study is that it kind of shifts our mindset, right? We're not just thinking about, oh, it's a Central Africa only disease. The potential may be there in other places. So wherever these species occur, we have potential interface between people where another spillover event could potentially happen. And how do we put policies in place to then try and stop that before it happens, right? Is the next step of that. And so if you add on this finding that we have from the species in Bangladesh, which is Rosetis leschinalti, a pretty cool bat, related to an African species, Rosetis aegyptiacus, which is also found throughout parts of the Middle East. It's the Egyptian fruit bat. So they're the same genus, they're related species. And so not surprisingly, actually, Rosetis in Africa is actually the main reservoir for Marburg virus that we know of. And we found evidence for potentially other filoviruses circulating out there in Asia where its sister species occurs. Right, so we can actually think about sort of the biogeography of disease as well, and the potential distribution of these viruses in the natural world. And so this is from that review paper that we wrote, and it just kind of shows you very crudely that mapping out these bat species ranges across the landscape. But this identification of what species is, I don't want to say to blame, but might be responsible for carrying a virus is not trivial, right? And it takes a lot of collaboration, multidisciplinary work, building teams, working with field biologists, people that know how to identify species, people that know how to catch animals, veterinarians. So we do this work, and this is from a training that we did in the Philippines for Ebola arrestin virus investigations that they were doing. So I remember arrestin's the one that came over on the lab macaques. Well, we were out there trying to figure out the source of the virus, where did it come from. So we haven't published this all yet, but another group did find some evidence in bats from the Philippines. And so we were out there catching bats, but not only doing that, we were trying to train up a pretty large group of local people to do this work themselves, right? We don't want to be flown into every situation. The idea is to build up the local capacity to do this kind of research at a country level. So fairly gruesome picture, I apologize. This is a picture I took in Vietnam. People do eat bats, right? And so there's been a lot of talk of it with the current West African outbreak because bushmeat consumption is huge there. People eat bats. There's been some epidemiological links between bats and Ebola outbreaks, but nothing super conclusive in my opinion. Marburg virus, several people have contracted Marburg virus by entering into caves that are known to be filled with bats that carry this virus. So that's a pretty clear link. There's some gold mines and some caves where gold miners have started some of the Marburg outbreaks in recent years. And CDC has done some excellent work really looking at the dynamics of Marburg virus and those bat populations. So it seems pretty solid, but again, we don't really know that single point in time where the human got it from a bat, and it's really hard sometimes to have that solid piece of evidence. But we know that this sort of behavior, barehanded chopping up animals, right, is probably not a good idea. It's a pretty risky behavior if that animal carries Ebola virus. And we're going to hear more about primate work later, so I don't want to scoop this. This is a picture I got from Billy Karish. It's from WCS. This is a mandrill, a dead mandrill, right, that this bushmeat hunter is taking out of the forest in West Africa. Obviously very intimate contact with these animals when you're hunting them, opportunity for spillover. And it seems like a lot of the spillover events have been linked to bushmeat, and especially through primate, the contacts with primates, and we're going to hear more about that. And I think I just want to make the point, and I think Damon, you'll probably make it later, is Ebola is not just a human disease carried by wildlife that affects us, it also impacts animal populations. So there's a nice study that found that in an outbreak in 2002 and 2003, estimated over 5,000 lowland gorillas died from Ebola, right? So this virus can spread in wildlife populations like it's spread in human populations, and can have a big impact on ape conservation. Now, I put these talking points together just largely in part because I want to avoid backlash, right, where people start blaming wildlife for causing these diseases. It's not the bat's fault, it's not the primate's fault. No one's saying go out and kill all the gorillas and chimpanzees because they carry Ebola, right? But some people are saying go out and kill bat populations because they might be carrying Ebola. And we don't want to do that because that's not the solution. And so this is an attempt to sort of, at least if you guys start talking with your friends about this, well, what are some talking points? First of all, the current Ebola outbreak is a humanitarian and public health disaster. It's human-to-human transmission. The genetics, the molecular genetics are very strong. It is probably one spillover event, right? Linked to one event, and it's spread in the human population. So it's not currently jumping in from animals and sort of fueling the fire. It was sort of a one-time event. And Ebola is a zoonotic virus. We know that. It's an animal wildlife virus that spills over into the human population. And while it's a rare event, it's a very high consequence event. And there's strong evidence, as I showed you, to suggest that several fruit bat species are natural reservoirs or natural hosts for these viruses, and potentially other related viruses, some of which we haven't yet discovered, no doubt. And we know that apes and other mammals are involved in that transmission cycle, and that people often get it through hunting bushmeat or handling animal carcasses that may be infected. But there's a lot we still don't know about that link between bats and primates and other other mammals that may carry Ebola. But what is clear, I think, is that human contact with wildlife is really the driving factor here, right? So it's through bushmeat hunting and other activities that are putting us into close contact with some of these animals. Well, if you're doing investigations, you should wear your personal protective gear, right? Your mask, your gloves, your respirator, like I showed you we do. But certainly, most people aren't out there just doing scientific handling of animals, right? They're out there, they're the big bushmeat trays, so they're eating animals. So we need to think about behavioral changes to sort of address that issue. I believe that these events can be prevented, and this is very strong belief we have at EcoHealth Alliance, that we can prevent spillover. We just need to have the understanding and the tools in order to do it. And culling of bats, or killing of wild populations, right? Culling is sort of a nice word for exterminating a wild population, is going to make matters worse. And there's actually a really nice study that came out of CDC with Marburg virus recently that actually showed that the prevalence of Marburg virus went up, increased after they culled the population. So basically what happened is bats from other areas probably came in and recolonized, they may have been susceptible, and there's actually a spike, there's a greater prevalence of that virus after they tried to cull and get rid of the population. So killing bats, definitely not the answer. It's dealing with our interface, right? Maybe stop eating bats, or if you're gonna eat bats, maybe do it in a safer way. So can we, at a landscape level, predict these emerging events? This is from that Nature paper, but sort of an updated model that we put together, looking at globally at hotspots for emerging infectious diseases, for disease emergence. And this is a combination of both understanding the wildlife distribution, but also the human population density, human behaviors, and ecological factors. You can kind of put all these things together into predictive models, and you can get across the landscape say, well, actually, this is a sort of a hot area for disease emergence. Oh, and look, West Africa popped up in that predicted map too, and this is now several years old, so we didn't fit the data after the fact, right? So you can, at least at a sort of crude level, predict this, but the real question then is, what can you do to prevent it? And so this is from some work we did with Nipah virus in Bangladesh, and with our colleagues at ICDDRB, which are a local organization in Bangladesh, did some really nice work to look at the ecology of Nipah virus. And we had Peter Daszak, who's my boss, president of EcoHealth Alliance, had 2 successive NIH grants to look at the ecology of Nipah. And we found out some interesting things. Well, we found out the exact interface where bats come in and feed on this date palm sap that people collect. This is a vessel to collect sap. And then people drink that sap in the morning. Well, bats are coming in and feeding on it all night long. They're urinating and defecating in this pot, and people are drinking that. It's like maple syrup. It's like a nice treat in the morning. Well, what can we do to prevent that? Well, really simple, really low-cost bamboo skirt that you put over the pot actually can exclude the bats and keep them from contaminating that pot. Right, so this is not a multi-billion-dollar solution. This is a 50-cent solution. And how can we get these kind of measures and implement these at a global scale once we do the research and we know what is the interface, what animals are carrying these viruses, how are people getting them, right? You kind of have to put the whole ecological story together in order to prevent. So with that, some funders, and thank you for your attention. I look forward to the discussion later.